My name is Nissinen and I'm working at MTT Agri-Food Research Finland, and I have been working with carrots and carrot psyllids for a long time. And then 2009, in the end of the year, it was discovered that Candida Liberibacter solanacearum is associated with carrot psyllids and the psyllid affecting carrots. And our latest paper is about the carrot psyllid feeding and bacterium teeter and their effect on the carrot yield. And in this paper we discovered that psyllid males make much less damage to the carrot root than carrot psyllid female feeding. The carrots were exposed for 3 days, both by females and males, and the effect of carrot root by male feeding was basically nothing. So there was no reduction in the yield or in the root size. While in female feeding, the effect was dramatic. This was pretty surprising. And we also discovered that the symptom of the bacterium is correlated. So bacterium teeter is correlated with the number of discolored leaves on carrot. And my colleague did a study of electron microscopy, and we got really nice pictures where the bacteria are pouring out of the phloem cells. So basically the bacteria block the phloem cells when their infection is developed far enough. So and that leads to reduction of the size, because the sugars are not transported from the leaves to the root. So, and then following study I did in UK in collaboration with my colleagues in Vera. We studied with an electrical penetration graph carrot psyllid feeding. So basically this device makes an electric circuit between the psyllid and the plant. And it makes different oscillations, so different kind of waves, depending in which plant tissue the psyllid is penetrating. And we wanted to know that if there is any difference between male and female feeding and between the feeding behavior on carrots and potatoes. Because the question is that are they able to transmit the bacterium in the potatoes? Even the carrot is the main host, and they reproduce there. So potato is not listed as a host of carrot psyllid at all, and they will die within a few days in potatoes. And basically we found out that both females and males spend more time in start of the penetration and psyllid in parenchyma, and in transition to the phloem tissues, and phloem ingestion and salivation on carrots than on potatoes, which was expected because carrot is the host plant. But we found one male which was able to penetrate to phloem in potato. So this suggests that about 8% of the carrot psyllids would be able to penetrate in the phloem, but the time they spent in the phloem feeding event at one time, or that one individual spent in phloem feeding event was on average 47 seconds, while in carrots, the males, feeds or salivates in phloem 15 times longer, so 12.2 minutes. And if we compare this with the females, so females were not observed phloem feeding on potato at all, but females spend more than double the time phloem feeding on carrot than males. So 25.7 minutes. And if we compare this time to the one which the male used penetrating in the phloem, which was 47 seconds, that's much longer, the feeding time in phloem in carrots, both males and females carrot psyllids. So basically it is possible that some individual of carrot psyllid would be able to transmit the bacterium to the potato, because some individuals do reach the phloem, but the duration of feeding event is very short. So it's unlikely to be an efficient vector